best way to go through the study guide is to bring up your notes. So I'm going to be doing that as we go through the study guide.
I'm trying to find my own.
7.4 in your book is the first part of Unit 4, and it's all about transport. So the first question on your study guide says, what is the difference between facilitated diffusion and diffusion? Remember that diffusion is a passive mode of transport, so it goes from areas of high concentration to areas of low concentration. The same thing happens with facilitated diffusion, except you need a helper protein in facilitated diffusion. Because it's passive, it does not require any energy. Diffusion keeps occurring up until equilibrium is reached. Okay, so there's movement until we reach equilibrium. Osmosis is the diffusion of water, so it's still passive, still requiring no energy, still going from high to low. So the three examples of passive transport we had were diffusion, facilitated diffusion, and osmosis. What direction is the natural concentration gradient? High to low. Very good. Remember our sledding hill. Okay, so it's really easy to go down the hill, but if we have to go up the hill, what does it require?
More
Energy. Alright? So in active transport, we're going against the natural concentration gradient, and we're going from low to high. We're going up that sledding hill, which is much more difficult. We have different types of solutions, hypotonic, hypertonic, and isotonic solutions. And the way that you name them is by looking at the concentration of water. Remember, water goes from high to low. Iso means the same or equal, so the amount of water molecules inside the cell is equal to the concentration outside the cell. In a hypotonic solution, water is going to move into the cell. Remember, hypo means lower or below. And we can remember what the cell looks like by remembering hippo, because the hippopotamus is big and round. Hypertonic, the prefix hyper means above or in addition to, so the higher concentration of solutes are in the cytoplasm. So water is going to move out. What does the cell do?
Stop.
It shrinks. Very good. If we need to transport something really large that can't get through the membrane, we're going to have to use endocytosis and exocytosis. Endo means to bring it into the cell. Exo means to put it out of the cell or to exit the cell. Alright, next we're going to talk about photosynthesis and respiration. Remember that cells need energy to be in the form of ATP, adenosine triphosphate. So first, let's look at photosynthesis. We're taking in carbon dioxide and water and sunlight, and we're making glucose and oxygen. There's two phases to photosynthesis. Phase 1 requires light, and it happens on the thylakoid membranes. Thylakoids are sacs filled with chlorophyll, which is the pigment that absorbs the light energy. In between all of the stacks of granum, we have stroma, which is where phase 2 takes place. Phase 2 is known as the light-independent cycle, the Calvin cycle. Remember, you can always go back and watch the videos that are associated with the unit. So now for photosynthesis' opposing process, which is cellular respiration. So if you look at the reactants of respiration, they are the products of photosynthesis. So in respiration, we're going to start with glucose, use oxygen, and we're going to produce CO2, water, and ATP. There's 3 steps to cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain. The first step is anaerobic, meaning it does not not require oxygen, and the second two steps are aerobic, which means it does require oxygen. Step 1, glycolysis happens in the cytoplasm. We're going to start with the glucose molecule and break it down into 2 pyruvates. If there's oxygen available, those pyruvates move into the mitochondria and perform the Krebs cycle. We get 2 ATP from glycolysis, we get 2 ATP from the Krebs cycle. Now it's time to really cash in the third step, the electron transport chain. We're going to capture all those free floating electrons that have been generated, and we're going to put them through the proton gradient and generate up to 34 ATP. If we don't have oxygen available, those pyruvates can't move into the mitochondria, and we do anaerobic respiration instead. The process that follows glycolysis is going to be fermentation. Alright, that's all for Unit 4.